Hello everyone, my name is Dr. Beth Williams. I am a lecturer in microbiology at the University of East Anglia, and I was also actually a student here just over 10 years ago, and I studied biological sciences. So today I'm going to be talking to you sort of setting up the topic of antibiotics and antibiotic resistance, and this is going to be a sort of pre-lecture to the Fighting Against Resistance, the Challenge of AMR session that I will be doing live. So this video is just a short presentation to give you some of the kind of foundational information and the background so that hopefully we can hit the ground running in the session. So today we are going to be talking about the grand challenge of antibiotic resistance. So we're going to start right at the beginning. So what are antibiotics? How do they work? We'll go into a brief history of antibiotics and how we came to use them and rely on them so heavily. And then we're going to talk a little bit about antimicrobial resistance and why it's spreading so fast. And then we're going to end with some questions that you can go away and think about and have a little read around if you're interested. And we will discuss those further in the live session. So first of all, what is an antibiotic? Well, I'm sure we have all in our lives, unless we're very, very lucky, been on a course of antibiotics. They are things that we take to fight infection, and the very name antibiotic means that it's something that opposes life. And these are kind of in the group of what we call antimicrobials, but they differ from cleaning products like disinfectants and antiseptics that are used in kind of medical preparation for surgery, because instead of just topical or surface applications, they fight infections within the body. So that's kind of the difference that antibiotics have. And they also specifically fight some organisms. So with antibiotics, we usually refer to bacterial infections, but we also have antifungals that can fight fungal infections. But the important thing is that they can selectively attack certain organisms without causing too much damage to the host. Although, as we all know, sometimes they can have some pretty nasty side effects. So antibiotics can be synthetic compounds, and especially the more recent antibiotics that we are using, partly because of technological increases, tend to be synthetic compounds, so compounds that we have made in a lab. But the majority, and certainly the earlier antibiotics, are all ones that are already being made by living microorganisms naturally in the environment. And one of the big contributors to these, what we call natural products or secondary metabolites, is Streptomyces. Now, Streptomyces is a really fascinating bacteria because it behaves in quite a different way to how we expect bacteria to behave. It doesn't divide by what we call binary fission, where the bacteria just splits down the middle and doubles each time. It actually produces spores, and it also produces a compound called geosmin, which we associate with the sort of damp smell of earth, which is actually a very pleasant smell. But one of the major reasons it's so interesting and studied very widely is the fact that it has a huge genome. It has loads and loads of extra compounds encoded in its genetic information, and that's what we call secondary metabolites. And it's actually has produced around 2/3 of all known antibiotics, many of which we are still using today. It's a very interesting organism, and actually the Norwich Research Park is kind of leading on Streptomyces research. There are a lot of brilliant minds working on it still to wring all every last secondary metabolite out of its genome. As well as antibiotics, it also produces compounds that we have used as anti-cancer treatments and also as herbicides. So a hugely impactful organism, and it also looks pretty funky on the plates, as you can see. So we're going to briefly talk about how antibiotics work. In the live session, we will have a little bit more of a discussion of sort of the things that antibiotics might target and how they might attack selectively bacteria. But essentially, we divide antibiotics into two groups. So some antibiotics we call bacteriostatic. So as the little diagram suggests, this means that they stop bacterial growth. So instead of bacteria doubling and causing this kind of exponential curve, growth is stopped. And they can do this through a number of ways that we will have a think about and then talk about in our next session. And then other antibiotics are bactericidal. And this means that they kill the bacteria. There is no more growth, they destroy it. So I said that microbes are the main source, microorganisms, not just other bacteria, but also fungi and some other slightly larger organisms. They make a lot of antibiotics. And why would they need to do this in just the normal environment? And the simple truth is that it's a very brutal environment out there. Everything is just really struggling to stay alive. Other things want to eat them. They want to consume all the resources. There might be difficult conditions like high temperatures. There may be a limited amount of food or maybe a limited amount of nitrogen. There's lots of things that bacteria need to compete for. And so competition for resources, one of the best ways to make sure that you get all the resources is if you kill all the opposition. So that's one of the main reasons that microbes produce antibiotics. But it can also be as a defense against other microbes that might be trying, kill them first before they can kill you. But because of this microbial war that's going on in the environment, we have managed to take advantage of that and selectively pull those antibiotics out. So it's been widely agreed that the discovery of antibiotics is one of the most important milestones in modern medicine. They have transformed the medical field. They have taken diseases that were once a death sentence into just an easily treated inconvenience. Things like a small cut or maybe an insect bite or an animal bite, very simple things. We can now just go and get a course of antibiotics, but years ago that would have been the end. So not only are they amazing for dealing with those kind of infections, but they have also led to the advancement in many other medical fields. So things like organ transplants, cancer therapies, treatment of kind of chronic diseases, any kind of open surgery. Then antibiotics are very good for fighting infections, or in the case of things where you have cannulas or internal instruments, antibiotics have made all of these medical leaps forward possible. So where did it all begin? We're going to use this QR code to go to a Padlet, and this has just the kind of layout of a brief history of antibiotics. Some of the names you might recognise, but I'm sure there are some that are perhaps not so well known. Okay, so as you can see here, that QR code has taken us to a Padlet that is called A Brief History of Antibiotics, and you'll be able to access this Padlet whenever you scan the QR code. And you can see here that we have laid out the timeline of antibiotics starting from the late 1800s, and I've just picked out some key moments in the story of antibiotics, all lined up here, ending in the 1970s. And so we're just going to go through each of these points and talk about why they were very significant points along the story. So in our brief history of antibiotics, there are quite a few points before even the 1800s that I could talk about, because ever since the idea of kind of tiny, what they called animalcules, but small, invisible organisms, so germ theory. Ever since that idea came out, we as scientists have been captivated by the idea of microbial interactions and how we might utilise those in treating different diseases. But the first kind of significant, what's often called the first antibiotic to be isolated, is this one by Bartolomeo Gossio, and it's mycophenolic acid, and it was actually also identified from the fungus Penicillium, and it was discovered that it kills bacillus. And so this is kind of the first ever antibiotic to be fully isolated and kind of characterized. And then we have, moving on to 1909 with Paul Ehrlich, and he kind of collectively theorized this idea that there are these substances that could selectively kill bacteria without harming other cells, other human cells. And in his work, he also synthesized a compound that was a very effective treatment for syphilis, which was quite a big deal at that time. So by 1928, we get to sort of the father of modern antibiotics, Alexander Fleming, which I'm sure you all recognise his name, because he kind of made the big discovery of penicillin. And it was actually by accident. So he had plated some bacteria on these agar plates here. This is an example, it's not a picture of the one that he actually did. But so he plated these, he went away on holiday to Suffolk actually, and then he came back and discovered that it had not been properly covered and that there was this fungus growing all over his plates. And as a microbiologist myself, I can testify that that does sometimes happen. But luckily, unlike what I would have done, he did not throw away the plate and pretend it never happened. And that's because he noticed this zone of clearance or zone of inhibition around the fungus that was inhibiting the growth of the Staphylococcus bacteria. So he theorised that it was producing some sort of compound that was preventing the bacteria from growing. And later on, he identified it and purified penicillin. It did take some time before we were able to test penicillin in a clinical situation. So Ernst Chain and Howard Florey were able to determine the structure of penicillin in 1939 and purified it at high enough amounts that they could test it first in mice and then in humans and confirm that it could be used to treat infections. Now, their methods might not pass muster by today's standards and regulations, but luckily penicillin didn't have any particularly horrible side effects, and so they could move along to getting it ready to be released in the next couple of years as a functional antibiotic. So there were other people also working on antibiotics, and Salman Waxman, he was one of the first to, although the word antibiotic had been used in previous times, he was the first one to properly use it in a medical sense and refer to these particular compounds as antibiotics, and obviously the name stuck. And across his time as a scientist, his lab together isolated over 20 antibiotics, and one of the ones that was most famous was identified with Albert Schultz and Elizabeth Bougie, and that was streptomycin, which, as the name might suggest, was isolated from the bacteria Streptomyces, which we just talked about. This was a really big deal because it was the first antibiotic that was actually able to fight tuberculosis, which was one of the biggest killers at that time. So as I said, penicillin was released a couple of years after it was purified. In 1943, it was officially used to treat infections, and by 1944 in World War II, it was essentially the Allied secret weapon because it was able to save so many thousands of lives. Those who were injured in the field and then in the hospitals throughout Europe, those places were breeding grounds for infections, and penicillin was able to save many a soldier that otherwise would have died. even after the kind of obvious release of penicillin, there were still advancements to be made. So in 1947, Mildred Rebstock found a way to synthesize chloramphenicol synthetically, and that led to being able to mass produce it, and her techniques were very revolutionary at that time. And then kind of from the 1950s all the way up until the 1970s, we have what we refer to as the golden age of antibiotics. And you can see in this figure here how many different ones were found. We call the kind of groupings of antibiotics antibiotic classes, and those are the groups where they all kind of do a similar thing, attack the bacteria in a similar way. But we have hundreds of antibiotics being identified in this time period, and just unfortunately you can see at the end here where the numbers start to drop off, and we'll talk about why that started to happen in a little while. So we've talked about all the kind of exciting parts of antibiotics and their discovery, but unfortunately that's not what this talk is all about. We are talking about antimicrobial resistance, which we tend to shorten to AMR. And bacteria are just really, really good at developing resistances, and you can see in this figure that we have antibiotics, the dates when they were introduced, and then here on the side you can see how quickly we identified resistant strains of bacteria. So this R means resistant. So for example, methicillin, it was introduced, and when we say introduced, we mean it was actually being used in 1960, and by early 1962 we already had methicillin-resistant staphylococcus, so MRSA. So it only took 2 years for those strains to first be isolated, and that means although we identified it then, they could have even potentially been around before then. In particular, penicillinase producing bacteria, so penicillinase is one of the methods that bacteria use to evade antibiotics, was actually identified in a staphylococcus 3 years before penicillin was even introduced into widespread use as an antibiotic. So obviously AMR, antibiotic resistance, is a really, really big problem. It's something that has been identified by the WHO. They have declared antimicrobial resistance to be a serious threat in 2014, although there were lots of warnings about it before that time anyway. Today, 700,000 people die each year from multi-drug resistant bacteria. So that's not just one antibiotic they're resistant to, but we're getting bugs that are resistant to huge swaths of antibiotics, and some of them are resistant to all the ones we know might work on them, which is very serious. And there's thoughts. So we've gone through our pre-antibiotic era, we had our golden era of antibiotic discovery, and there's fears that we are now moving into what we call a post-antibiotic era, where we are struggling to keep up, partly due to the amount of time that antibiotics take to develop versus how quickly AMR is spreading. And there are some predictions that the number of deaths will rise to upwards of 10 million in 2050. So this is a very, very serious problem, and it's something that we need to be funding and thinking about and talking about and spreading awareness of. And so that's what this presentation is about. But it's not all, our talk isn't going to be all bad news. There are things that are happening and potential solutions to this, and that's what we're going to be focusing on in the next session. So I mentioned that we had our golden age of antibiotic production that kind of petered off. And in fact, the last time we found what we call, so we group antibiotics depending on the way that they impact bacteria. And the last time that we found a new group was in 1987. And although we have identified some after that, they've usually been either modifications of previous ones or just different variations within the same group. We just aren't developing them fast enough. So why aren't we finding as many as we used to? Well, the first reason is the fact that it's very hard. We have what we call the rediscovery problem, where the bacteria and fungi that we're able to culture in the lab, we've already identified all the antibiotics that they are naturally producing. And so now we've got to look at new methods or ways of getting them to produce new antibiotics, which is very difficult and time consuming. So they have to go through a lot of clinical trials in order for an antibiotic to be released into the public. Obviously, we need to know what impact those antibiotics are going to have on our bodies in short term and long term. So in the next 10 years, we might only be able to release another 15, and maybe 4 or 5 of those will be broad spectrum antibiotics that can really have an impact on the high risk infections. It's also very expensive, and unfortunately pharmaceutical companies tend to invest in developing more lucrative drugs that aren't going to get defunct in a couple of years once there's a resistant strain to them. So funding is another reason why we have this lack of antibiotics. So finally, we kind of talked about the lack of new antibiotics being one of the reasons that resistance is so high. But why is it spreading so fast? And so there's a couple of other big factors that are part of this that we will talk about in the next session. So bacteria are just unfortunately really, really good at adapting to their environment. And bacterial evolution is a really key reason for this resistance to develop. But in terms of the spread, antibiotic misuse and the ways that we have made the situation worse are also something that we're going to talk about. So something to think on before the next session. So those 3 are the kind of major reasons that AMR is kind of happening and spreading and becoming such a problem. So I said that we in 2014 talked about it was announced that AMR was a huge problem, but it's been something that we were warned about. So Alexander Fleming, in his Nobel Prize address in 1945, talked about the issues about how easily penicillin how easily bacteria can develop resistance to penicillin. He talked about the danger that human activity can have, and the kind of how we can exacerbate those problems. And he was, of course, right, and we should have not rested on our laurels of the antibiotic golden age and stayed vigilant. So that's about it for this video. For the next session, there's a couple of things that I would like you to think about. So one of them is, how are bacteria so good at developing resistance? What is it that makes them able to evolve resistance and adapt to our environment so quickly? Also, how have we as a society made it worse? What ways have we misused antibiotics? And as a kind of flip side to that question, what is it that we can start doing now to help slow the spread of antimicrobial resistance? And also, on a hopeful note, what kind of alternatives, new ideas, new ways of finding antibiotics might there be, and that we might talk about in the following session. So thank you all for watching this video, and I look forward to interacting with you in the future.